Well, welcome everyone to today's Tech Talk. It's brought to you by the MPCCC, the Monash Partners Comprehensive Cancer Consortium. My name is Chu. I'm a cancer geneticist and bioinformatician in the Department of Medicine at Monash University, and I'm the convener for the MPCCC Tech Talk series. To begin, I would like to pay our respects to the traditional custodians of the lands from which we join together today and acknowledge their elders past, present, and emerging, as well as any members of the Aboriginal and Torres Strait Islander community who may be among us today. So to set the scene, the MPCCC Tech Talks aim to provide an informal and interactive forum for our professional research community to discover and discuss the latest cancer research technologies and how they can be applied in practical ways to support our research. The Tech Talk format offers plenty of time for Q&A, and we really encourage you to tell us about your experiences, your key learnings and tips and tricks, as well as any potential challenges and limitations you face in your research. So today, I'm very pleased to welcome Associate Professor Sefi Rosenbluh. Sefi is a mid-career researcher that has made major contributions in functional cancer genomics and our understanding of cancer dependencies. After completing his PhD at the Hebrew University of Jerusalem in Israel, he moved to the Broad Institute of Harvard and MIT as a postdoctoral fellow, and later as an instructor of medicine. In 2017, Sefi joined Monash University, and now, in addition to heading a research lab, he's the director of the Monash Functional Genomics Platform. So in this seminar, he will discuss the use of CRISPR, base editors, and scanning mutagenesis screens that enable identification of new drug targets, mechanisms, and pathogenic variants. So for the presentation, and I think everyone's already muted, but please remember to stay muted and turn off your video to minimize disruptions. And we'll have questions after. So thank you, Sefi. I'll hand over to you now.
Thank you, too. Let me just share my screen. All right, could you all see my screen?
Yes, we can.
Okay, good. All right, so today I want to talk to you about, my hat as a director of a platform, as Tu said. I'm going to try not to really go much into the biology of things, but more talk about the different methods we have in the platform, some of them very established, some of them newer methods that are being already developed. And I'll try and give the different advantages, disadvantages of each of these approaches, and maybe some examples of how they are used. So we'll start with the motivation for everything we do in the functional genomic platform, which is to connect between genes and variants to phenotypes. And this could be in cancer. I mean, in this instance, we're talking about cancer, but this obviously could be done for also non-cancerous phenotypes, and I'll show some examples of that. And the idea is that we want to perturb or introduce genes, so either overexpress, suppress genes, introduce variants, delete variants, and then measure phenotypes. And the motivation of this is because, for example, if we want to find new drug targets, if we want to find new combination therapies, if we want to find variants that are associated with the different forms of risk of cancer, all of those are drug resistance. We'll talk about all of these kind of approaches and how they work. So I'm going to show a lot of different technologies. And the one important thing that underlies all the technologies and everything we do in our platform is this idea of pooled genetic screens. So I'll just explain here shortly how this works. So the basic idea here is that we create a library of lentiviruses. They could be sgRNAs if we're using CRISPRs. They could be ORFs. They could be shRNAs. They could be a wide variety of things. So we call these the perturbogens. So these are viruses. We could create them at very high throughput, relatively not expensive. And they could either overexpress, suppress a gene, introduce a variant, do all these things. and we create them as a pool. So we have one pool, one bag that has all of these different viruses, each one encoding a different reagents or perturbing a different gene or variant. And then we infect this into a cell of interest. And we do that so that we have one perturbant per cell in the culture, and then we apply a selection pressure. So selection pressure could be a drug, could be just growth, if you want to see what inhibits or enhances growth. It could be a reporter, and I'll show different examples of this as we go on. But the idea is that the selection pressure will cause a change in the distribution of the cells. So here you see in this example, at time 0, all the perturbagens are presented at a similar frequency in the population, but after we added selection pressure, that has changed. And we could measure it by just extracting the genomic DNA and sequencing. So we use sequencing for counting. So in this example, this is what this would look like, that in time 0, all the guide RNAs in this case are present in an equal proportion. But then after we applied a selection pressure, we see some guides RNAs are depleted. So these genes were disadvantages for the selection pressure, or we see genes that had an advantage for this selection pressure. So that underlies all the screens that we do in the platform. I'll first start by talk about loss of function screens. So those are probably the ones that we do the most in the platform. And here we use one of 3 different methodologies, and I'll talk of the advantages and disadvantage. So the goal of these loss of function screens is to inhibit the expression of a particular gene of interest and then measure a phenotype. And we could do this in one of 3 ways. So either using CRISPRs, and we use 2 different methods of CRISPRs, and I'll talk in a minute why, what's the difference between them. In one of them, we use wild type CRISPR, in which we cut genes in the DNA and knock out genes. The second one is CRISPR-I, or CRISPR inhibitory, in which we use a catalytically inactive Cas9, it's called dCas9, in which we could direct to promoters of genes and inhibit transcription. And the third one is old RNAi, in which we direct a hairpin that targets a particular gene of interest. So we're targeting the mRNA of a gene and inhibiting this. So I'll start with the history of this. So the history of this obviously was using shRNA screens, and this is my formal work, in which we could use these shRNA screens and use the same kind of methodology that I showed you before, these kind of pools, and find genes that are essential for proliferation of a particular cell. Now, what you could do in this example here, what you're seeing is we were looking here, we were interested in genes that are important in the context of Wnt activity. And here you're looking at Wnt, we could differentiate between cells that are Wnt active or Wnt inactive. Every row here in this heat map presents a different gene. And here we're looking for genes that are essential for this population of Wnt active cancers. And we found some validated and showed this new kind of pathway that we found is important for these Wnt-driven cancers. But what is not shown in this, and it happens in a lot of shRNA screens, is this idea of problem of off-target shRNA effects, which has been a major issue with the use of shRNAs. So what are shRNA off-target effects? And this word has been used quite a lot, but just to quantify it, this is what we mean when we talk about an off-target effect. So in this example here, you see that for our particular phenotype, we have gene A and gene B and so on. So each gene is represented by 4 different shRNAs. And each shRNA across a particular cell line has a profile. Right, so in this case, blue means the cell is dying when you introduce this shRNA. Red means it's surviving. And in this example here, if you look at these, for example, in gene A here, you could see that shRNA number 1 and 2 are very similar to each other, and number 3 and 4 are very dissimilar. They're doing kind of something different. So the first question that you want to ask with this, and then we've done this when we have done after we've done a lot of these screens and hundreds of cell lines, is to ask. So these shRNAs, 1 and 2, which we call sister shRNAs that are aimed to target the same genes, how many times do they actually target that same gene of interest? And when you do that comparison, so here you're looking at the correlation between shRNAs that target the same gene, or random shRNAs, so a kind of random shRNAs here in blue, and these are shRNAs targeting the same gene, you could see that they are pretty much random. That means that the shRNAs that are designed to target the same gene are actually many of the times doing something very different. And that is the major effect of shRNAs. And if you ask two shRNAs that were designed to target a similar gene, what are they similar to? The answer is that there's seed region here, an shRNA seed region. It's 9 nucleotides here or 7 nucleotides, sorry, that probably have this microRNA-based effect, and two shRNAs that were actually designed to target completely different genes, but share that seed sequence, have a very similar phenotype. There's an algorithm developed by Aviad Cherniak at the Broad, which tries to correct for this phenotype, but obviously this creates a lot of issues with the data set. And that is the reason that it's not that we never use shRNAs, and sometimes we use them, but you have to be very careful, because every time you're using an shRNA, you're actually introducing two perturbations simultaneously. One is you're targeting your gene of interest, but the second is that you are mediating the seed effect, so this non-specific effect that is suppressing your gene. Still, in some cases, we still, for example, projects that we want to target non-coding RNAs, for example, still shRNAs is the best approach currently still available. But obviously, when CRISPR came out, we were very excited that we could use a different method to suppress genes. So the first method I'm going to talk about is the wild-type CRISPR, or CRISPR knockout, as we call it. In this approach, we use a wild-type Cas9 in which we target to a exonic region of a gene. It creates a double-stranded DNA break, and it continues and cuts and cuts and cuts until you get this indel or non-stop or stop codon. And in this experiment, we compared between CRISPRs and RNAi, and to look at how good they are. So when you look at positive controls, so this is using CRISPRs, and here we're using cancer cells with either mutant or wild type of these particular genes. What you see is what we see with shRNAs as well, and this is what you expect to see, that when you knock out these known dependencies, you could see that you get what you want to get. So this is kind of a plot that it's going by itself. This is a plot that we kind of typically see. And you're looking here on the y-axis is proliferation. So minus means cells are dying, plus means cells are surviving. And if you're looking at these positive controls, this is what we see, what we expect to see, that the mutant cells are dying once you introduce the mutations, while cells are not. But what really got us excited about this initially is when we looked at negative controls. So here are comparing shRNAs in red or CRISPRs in blue, using shRNAs or guide RNAs targeting non-human genes. So, GFP, RFP, or luciferase. So here you would expect them not to do anything. And that's what we see for CRISPRs. They're all centered around 0, that's what you want to see. But what you see for shRNAs, and anybody who's used shRNAs would know that you see many of them have these off-target effects. As I showed you before, these seed effects, which even though they're not supposed to do anything, they are doing things in the cells. So that got us very excited about CRISPRs. But I think we've learned from all of our previous mistakes. And what we know is that all of these methodologies, and these include 100% of the methodologies that I will show you today, all have issues. And the main issue that we see with CRISPR knockout, is this bias in genes that have amplified regions. So here's an example of a cancer cell line. This is a pancreatic cancer cell line in this example that has an amplification of chromosome 19. So this is the profile of the amplification of the copy number of this cell line. Here, you're looking at CRISPR guide RNA, so CRISPR scores. So each dot here represents a gene in the genome. Again, minus means killing, plus means surviving. And here you're looking at the profile. So here's for CRISPRs, or here's with RNAi. And what you see when you get to this region, suddenly all the genes in this particular region here, in this amplified region, become essential. right? You don't see this with RNAi, but you definitely see this very clear signal with CRISPR. Another way to show this is by just looking at the correlation between copy number and dependency. So irregard to the actual gene, once you have high copy number, what you see is a very strong dependency. And this is something we should have maybe thought of before, but what typically happens is when you have an amplified region, you have multiple copies of that piece of DNA, and then when you introduce the CRISPR, you get this very high amount of DNA damage, which causes non-specific cell death. And that is a huge issue that we see with CRISPRs, with CRISPR cutting, CRISPR knockouts, is that when you target amplified region, you get this non-specific killing, which has nothing to do with targeting the gene, but with introduction of DNA damage. That's why we went to another methodology called CRISPR-I. Now, CRISPR-I uses the same CRISPR machinery, but the difference is that CRISPR-I uses a dCas9. So this is a catalytically inactive Cas9 called dCas9. So the two active mutants are no longer active. It could still bind to DNA, but it can no longer cut DNA. And this is also fused to a transcriptional repressor called CRAB, and the idea is to inhibit transcription with this complex. Here, the comparison of the two methods that I talked about. CRISPR-I uses a catalytically inactive Cas9, and CRISPR knockout uses an active Cas9. So to look at the determinants of CRISPR-I, we used a 6-TG screen. So 6-TG is a compound that is toxic to cells that have suppression of HPRT1. And we compared the CRAB dCas9 or CRISPR-I to the wild type Cas9. So what you expect to see here on this screen, each dot here represents a different guide RNA. And what you'll want to see is that if this screen worked and you really got knockout of HPRT1, you will see more proliferation or rescue of the phenotype. And if you look at Cas9, you could see whether you're targeting exons or you're targeting the promoter of the gene, you could see a very nice rescue with almost every guide that we take. But when we look at the CRAB dCas9, you could see that when you target the exonic regions, nothing really happens, only when you target the transcriptional start site. And the way we kind of envision this is basically that the system is kind of like a train. So if the train is going at full speed, you can't really stop it anymore. So that's the transcriptional machinery. The only way to stop it is in where it starts. If you go to the first station and take out the conductor, that's the only way to stop the train. But the issue that we had with this is when we looked at this is that many of the guides, even when we target the transcriptional start sites, didn't work. So here is an example of targeting 34 cell essential genes, so genes that when you're knocking them down, you expect them to inhibit proliferation. And we do see a signal in the transcriptional start site, but many of the guides, even in the transcriptional start site, don't work. And when we asked what is actually happening here, what we realized is that our annotation of transcriptional start sites is not the greatest. And our initial approaches were using NCBI annotation of transcriptional start site, which are not very good. But then we started to say, maybe we'll use a different annotation of transcriptional start site. So we used this database called CageSeq, which is an experimental way to measure the transcriptional start site. And when we do this, what we found is that a lot of transcriptional start sites that may be annotated in NCBI are actually not correct. And here's an example of MYC, so not some obscure gene, but even MYC. The transcriptional start site of MYC in NCBI would be annotated right here. And this, if you look at CageSeq, so here's CageSeq, it has two peaks. And then this peak here is a smaller peak, so it's probably not the main transcriptional start site of MYC. And when we use CRISPRs to target this transcriptional start site, actually nothing happens to the cells, but only when we target this second transcriptional start site that NCBI did not annotate do we see suppression of cell proliferation. And this tells us is that this won't affect the protein coding sequence, it only express the 5' UTR, but that's why it's really important to we started using much more of this cage-seq methodology, and this is just looking this across a lot of genes. We could see that you only get a signal when you take the highest peak of these cell essential genes. So that is good, and based on this, we have a very good methodology to identify where to target and how to target genes with CRISPR-I. Looking at DNA damage, what is nice is that CRISPR-I, we don't see at all this approach. So here's doing screens comparing wild type Cas9 or CRISPR knockout to CRISPR-I. And what you can see in green here are the distribution of negative controls. So these are guide RNAs targeting non-human genes, and they center around 0 for both CRISPR-I and CRISPR knockout. That's what you would expect. But when you, and here are in blue, guide RNAs targeting core essential genes or common essential genes, the exons of them or transcription start sites of them, and you see what you expect to see, a shift to the left. That's what you expect to see. But when you target AAVS1, so this is a region not containing any genes of interest. So when you target that region, nothing should happen. And this is what you see. Here's the effect of DNA damage. So just by introducing DNA damage, you already see some shift in proliferation, which you do not see with CRISPR-I. But like I told you in the beginning, and this is a very important message for everything I say, there is no perfect methodologies, and every methodology has issues. And the main issue that we have found with CRISPR-I is this problem of bidirectional promoters. So about 10% of the genes in the human genome have this kind of structure in which you have two genes that shoot in two opposite directions. And if you target CRISPR-I to this promoter right here, it'll simultaneously suppress both genes. And that's why if you use CRISPR-I here, for example, for these two genes, it'll look like both of these genes are essential for cell proliferation. Although when you use CRISPR knockout, you could actually see that only one of them is actually essential. And that is a major issue that we find with CRISPR-I. The method is, the message that I want to say here is that probably any method you use, you should always validate, and it's probably good to use multiple methods to look at your approach. We typically find that CRISPR knockouts give stronger signals. That's why we like to use CRISPR knockouts, but we like to validate things with multiple different methods. Now, one thing that we have done in the past, just to show you why this is useful, is we could actually use CRISPR-I to look at alternative regulation of transcription. Because CRISPR-I only works at that promoter region, what we have screened is a library of CRISPR-I that target either canonical promoters or these alternative promoters that are gained in cancer. And we can find some of these new promoters that mediate the transcription and the translation of new kinds of transcripts that are important for cancer proliferation. So that's a use of CRISPR-I for finding something new. Another example is a screen that we've done together with Jian Li's lab and Zulu from his lab using CRISPR knockout. So here was a screen that was aimed at finding genes that mediate resistance to polymyxin B. So polymyxin B is an antibiotic that is very effective against bacteria, but is toxic to kidney cells. And we were looking for genes that when you knock them out would reverse that toxicity. So what we have done is we've taken our kidney cell and either treat with the antibiotic polymyxin B or a controlled DMSO, and we look for genes that mediate this resistance to polymyxin B. And this is what a screen like this looks like. So you could see here the magnitude, the log2 full change, and the significance or the p-value. What we found here is there were multiple hits, but these two hits were very clear winners. So these are two components or two parts of a potassium channel, which then they went on and validated and showed in different methods and different approaches how this potassium channel mediates the resistance to polymyxin B, which suggests a weight of a combination therapy and treatment to reduce toxicity of this channel. A third example that I will show is a screen that we did together with Sarah Jones's lab from the School of Clinical Science. And Sarah's lab is interested in looking at anti-inflammatory, and that's why they were interested to finding genes that regulate the transcription of this transcription factor, GLITz, or TSC22D3, which is important for anti-inflammatory signals. So in their screen, what we have done is we again put in our lentiviral library. Each cell has here one guide RNA, but here instead of looking at proliferation, we use FACS sorting with a GLITz antibody. So we could sort cells that have low or high level of this protein. And just to remain confidentiality, I didn't add any gene names here because this is not published yet. So I just put in kind of our controls to show you that this works. And then you could see here in this portion of cells that have low GLITz, our top hits are, as you would expect, the actual gene of interest and its binding partner, right? So that was nice to see that they are the top genes that score, so that shows you that the screen works. And obviously they found other genes that are of interest and they're validating and working on them right now. Okay, so those were CRISPR knockout screens in which we have, and we've done many, many screens with these now, and anybody who wants, you should come talk to us. A newer technology that we have just now started to apply is combinatorial screens. So combinatorial screens, meaning libraries that don't only suppress one gene, but suppress simultaneously more than one gene, typically 2 genes, but it could be even more than that. The reason that this is very useful is, number one, looking at paralogs. So about 10 to 20% of genes in the human genome have paralogs, meaning that you'll have multiple genes that do the same function. So if you just knock out one gene, you won't get a phenotype. The second reason for that is looking at genetic interaction mapping or combination therapy, which a lot of us want to use more and more of. How do we do this? Using typical CRISPR wild type, it's very difficult to do this. It's possible, but extremely difficult because you need 2 promoters and you have 2 guide RNAs that have a similar tracer sequence. That is very difficult to clone, very difficult to work with. But Cas12 is a different Cas enzyme, so which the advantage of Cas12 is that it, as opposed to Cas9, Cas12 knows how to process its own guide RNA. So what you could basically create is it has a very short tracer. It's called these direct repeats that it has. And we have a couple of different direct repeats that we could use. So we just create an array of guide RNAs that target whatever genes we are interested in. And the Cas12 itself knows how to basically cut these guide RNAs based on this direct repeat and use them to target a gene of interest. And here's to show you that this works very nicely. And here we are targeting BCL1 L1, so BCL-XL and MCL1. And what you can see, this is very specific. And also that when we target both BCL and MCL1, which are known synthetic lethal genes, we see cell death in this particular cell. So this method works. And here's a screen that we've done together with Ron Firestone and Hugh Gao from his lab. I'm not going to show many results, just again, looking at controls here. So here you're looking at a collection, and in this case, it's close to should be around like 100,000 different combinations. And then what you could see is how much are these combinations deviating from the expected phenotype, which is either synergy or epistasis. And what is nice to see here is that you see a lot of paralogs that score in this synthetic lethality or this genetic interaction or synergistic interactions between them. And that's now we're creating a library. So together with them, we're making a library of more than 1 million combinations. But this is something that could be used for both looking at interactions between genes or these paralogs. Okay, so I talked up to now about loss of function screens. And I'll go now to talk about gain of function screens. I'll try to go, I'm running out of time. I'll try and do this quick. So in gain of function screens, what we want to do here is overexpress a gene of interest. And we have two ways to do that. One is with ORF libraries, which we have not been using so much of. In ORF libraries, we have a library that has 16,000 ORFs in it. That causes a very dramatic overexpression of a gene. It's usually not very physiological. And some of the genes have in them all kinds of different mutations of sorts. So we have not been using quite a bit of it. The other method is using CRISPR-A or CRISPR activation. So in CRISPR activation, you use the same methodology that I showed for CRISPR-I. So that's the inactive Cas9 or the dCas9, but here it's fused to a transcription activator. So here you can see it's VP64, but we actually use one that, in addition to VP64, brings a combination of different transcription factors and results in robust activation of gene expression. The methodology we use in this method is very similar to what we use with CRISPR knockout. It's just with a different Cas9. I'll just show you here a screen that we recently did together with Dan Goff from the Hudson. So in this screen, what we did is we were looking for genes that mediate resistance to carboplatin in a small cell cancer cell line. And the idea is to overexpress genes and to see which one of them will induce resistance to carboplatin. I'm again not going to show any of the hits, but just the control hit, SLC7A11, which is a known gene that induces resistance to carboplatin, and this is one of our top hits in this screen. And they are following the rest of the screens. Okay. So those were the main methods that we use for suppression of genes, so either combinatorial, overexpression, or suppression of gene. And the next slides that I'm going to talk about is about variant screens. So in variant screens, we're interested in not suppressing completely a gene or not overexpressing a gene, but we're interested in introducing a variant, a mutation, and measuring the phenotype that it mediates. We have two ways that we could do this, either with base editors. So base editors are, again, dCas9, but here dCas9 is fused to a DNA modifying enzyme. And currently we have two different DNA modifying enzymes, ABEs or CBEs. So ABEs will convert an A to a G. CBEs will convert a C to a T. Right, so we have those different base pairs that we could induce. The disadvantage of these is that you are limited because you need to have the guide RNA PAM sequence, and it only edits at a limited window. The advantage is that you could tile across a complete gene, including introns, including exons, and you could look at the endogenous region, what happens when you edit that gene. Here's an example of a few edits that you could see that we could get like nice editing of these regions. The second method that we have is called scanning mutagenesis. So here we create a library of plasmids or ORFs that overexpress for a particular gene of interest. We introduced every possible amino acid variant in every location in the gene, and then create a virus and measure the phenotype. The disadvantage is that we are looking at exons, we're not looking at introns. We are overexpressing the gene, so it's not the same as a kind of the physiological expression of a gene. And we are missing things that might happen at the genomic locus that we cannot see here. So I'll just give an example here of a screen that we are still in the middle, but I'll show you some of the results of it, of CDH1. So CDH1 is a cancer risk gene. Individuals that have pathogenic variants in CDH1 or E-cadherin have a very high risk of getting gastric cancer and higher risk of getting also a form of breast cancer called lobular breast cancer. And the thing with CDH1 mutations is they're extremely rare in the population, so it's very hard to get enough individuals to do actually population genetics. And that is why if you look at the distribution of the mutations in CDH1, what you'll see is that only a small portion, about 11% of the genes in CDH1 have been completely classified as pathogenic, and actually more than 50% of variants in CDH1 are considered as VUSs, so variants of uncertain significance, and most of these variants of uncertain significance are misses variants. So most of the pathogenic variants are known nonsense variants, but a lot of the missense variants are not classified. So I'll show you here how we are using base editors in this. We're also doing it with scanning mutagenesis, but right now I'll just show you the base editor screens. So first of all, when we want to do this screens, we need to get a phenotype that we can measure. And in this case, what we did was look at DepMap. So DepMap is a map of cancer dependencies, and we found that two pancreatic cancer cell lines required CDH1 for survival. So in these cells, when you knock out CDH1, these cells die. And we could actually validate that in a low throughput method. When we knock out CDH1, these cells do not survive. So we chose PANC 0327 for doing base editor screens. And this is how the screen works. Once we create the library, we infect with the library. And again, we grow the cells for a while. So this is our selection pressure, and we're looking for the abundance of the guide RNAs. The guide RNAs that disappear would be essential for cell survival and not. So looking at the quality controls, our library also includes variants that introduce variants of splice donors or in pan-lethal genes. So these are genes that we know are essential for cell survival. And this is what we expect. And this is what we see, is that when we knock out these genes, you see a very dramatic decrease in cell proliferation. So that's very good. That says the screen works. If we look at CDH1 exon targeting sgRNAs, what you could see is the majority of them are not inducing a phenotype, which is good, and that's what we would expect. But within our cohort, within this ABE screen, there were 3 of the variants that should introduce a known pathogenic variant. And you could see right here that all of them scored very nicely in our screen. Usually guide RNAs that show a decrease in proliferation below a -1 log fold change, we consider as a very strong hit, and all of these scored as known as strong hits. And now we're looking at the rest of them to see what they are doing. An interesting finding, I'll just quickly say, that we saw out of this is when we're looking at intronic regions. So here's the structure of CDH1, and what you could see is that there's huge intronic region between exon 2 and exon 3. And what we found that there was we have guide RNAs in our library tiling across the entire gene, but what we find is a particular region here that had multiple guide RNAs that showed a very dramatic phenotype. We don't think that this is related to splicing. It could be, but because it's kind of far away from the exon, we don't think it is related to splicing. And we're now looking at how are these intronic regions maybe mediating CDH1 function. And this is very important because it opens up a whole new way of looking at these variants, because currently most of the genomic approaches are not actually measuring the intronic variants and what they are doing. So this is an important and interesting vignette that comes out from these kind of screens. Okay, so the last method I'm going to talk about is, I talked about methods for screening for variants, for screening for genes. The last methods I want to talk about is methods for annotating gene function. And this is something that we a lot of time miss in our screens, because we finish a screen and put a lot of effort in the screen, but then obviously what a lot of the work is to take that information about that gene and find out how that gene is working, how to target that gene, what to do about that gene. And that's usually a major challenge in all the screens, because we could get validated results, but to figure out what they are doing is extremely challenging. And for this, we adopted this methodology called CropSeq. In CropSeq, what we do is we use a library of guide RNAs. So this is a smaller library, not as large as a genome-wide, but you basically will take your hits on the screen or something like that. They are barcoded in a way that they could be read on a single-cell RNA-seq machine. So what we do is we infect our cell population with these guide RNAs, and then use single-cell RNA-seq to both identify the identity of the guide RNA and the global expression changes that occur after introducing that guide RNA. And this works very nicely. So here is seeing a pilot experiment that we did that we can inhibit the expression of the target gene of interest. And what we found is that some genes, and that's typical for single cell experiments, are detected at low levels. So just the transcript of the gene is not very robust. So you almost don't see really good knockout of the gene. But if you, for example here, if you look at Raptor, for example, a known component of the PI3 kinase pathway, so we don't really see good knockout of Raptor. But if we look at the gene expression changes, the signatures that are mediated by sgRNAs knocking out Raptor, they are consistent with a hallmark of mTOR signaling, which is what you would expect to see. Similarly, casein kinase 1 alpha, which we don't see very good knockout. When we look at the expression signatures, we look at axin 2, casein kinase 1 alpha is a known regulator of the Wnt beta-catenin pathway. We see overexpression of it. So that's to say that this methodology works very well, and we could suppress genes of interest and see expected phenotypes. Now, what could you do with this data set? So the first thing that we've seen with this data set is if you take genes that you use in this screens and you cluster them, you find known components of signaling pathways. So known signaling pathways tend to cluster together, and the idea is that you could find now new genes that are not part of these, that you did not know before, and supposedly, based on the clusters, think or hypothesize what they are doing. The second thing that we could do with them is we could use databases that we have and look at compounds that target this gene. So L1000 platform is a database of expression signatures mediated by hundreds and thousands of different compounds. So we could just take a gene expression signature that we get after knockout of a gene, input it to that database. It's very user-friendly and easy to do. And then we could find drugs that target that gene. So here's some examples that we have done. So here are known components that we could look at, either knocking down of p53 will give you MDM inhibitors, knocking down of PTEM will give you mTOR inhibitors. And then an interesting one that we found is knockdown of CREB-BP, which was correlated with vorinostat, an HDAC inhibitor, and we could validate that very nicely, that when you knock out this gene, cells become sensitive to vorinostat. So this is a way to at least try and connect between genes and compounds. The other thing that we have used quite a bit of CropSeq for is doing this connection between gene enhancers and target genes. So one of the major issues a lot in GWAS is that a lot of GWAS targets are in enhancers, and one of the things we want to do is find the targets of those enhancers. And what we do in this methodology, we direct a dCas9 CROB, so an inactive Cas9, to this enhancer, and we measure the changes in expression of genes that are downstream of this enhancer. So just to show you that this works, when we use this method, we could see control genes go down. But more interestingly, here's an enhancer that we have targeted. And in this enhancer, each line here represents a different guide RNA. And what you could see is that this enhancer here is suppressing the expression of 1 or 2 or 3 genes. And this was the major gene that we found that it's suppressing. So here you could see, and the height of this arc is the degree of suppression. And we could see that for this gene here, this enhancer was able to suppress all the guides, suppressed basically the expression of this gene, which we then went and validated in a lower throughput, and we show that when you target that enhancer, either by luciferase assay or by CRISPR-I, we could suppress the expression of this gene, showing that this enhancer actually targets this gene ATF7 IP, and we've together with other screens have published this a while ago. Okay, so with that, we're over time already. I will end. Just thank a lot of people that were involved. I didn't show all the kinds of screens we have. I hope this was clear. Thank my funding agencies. as well as different members that helped or did a lot of these screens. And if you're actually interested in doing a screen or want to discuss this, please contact either myself or Murray Manning. His email is right up here, our platform manager, and we could discuss how to do and engage with this. And that I finish, and I'm happy to take any questions.
Thank you very much, Sefi. So please feel free to unmute yourselves and turn on your video if you'd like to ask a question. Or you can also type them in the chat. Do we have a...
Hi, this is Antonella. Can I ask a question?
Sure.
Yeah. Hi, Sefi. So thank you very much. This was a great presentation and really brings everybody up to speed with the world of CRISPR. So that's very good. I have a question about the CRISPR-I. Because you said that you basically, so the way the CRISPR-I works is really to interfere with the transcriptional machinery.
Yeah.
And with maybe interacting with let's say inhibiting or affecting the transcription factor in the complex at the transcription start site. So you said that often you can also have bidirectional promoters, but I guess, with the CRISPR-I, you can also affect transcription start site that not just work in cis, but also in trans. And therefore, I'm assuming not the side effect or the unwanted effect might be even higher than the classic CRISPR knockout.
I think that's definitely true.
Is it possible? So then,
that's definitely true.
When it comes,
it's hard to measure that.
Yeah.
That's why so the bidirectional promoters are easy to measure.
Yeah.
Because they're very clear. Those like cis effects, I think you're very right. I'm sure that they exist. We just don't have a way to systematically measure them.
Yeah. And so do you still recommend doing a CRISPR-I together with the CRISPR knockout, or basically you do a CRISPR knockout irrespective of all the off-target effect that you get, and maybe test different guides? Even though the same guides will still give you.
At the end of the day, it's a matter of how much money you want to spend on doing the screen, right?
Okay. Yeah.
Typically, what we usually recommend is to do a CRISPR knockout screen.
Yeah.
Then validate with different methodologies your hits.
Okay.
Those are where we typically see the best hits. But if you have unlimited resources,
Yeah. So the CRISPR-I is not included in the CRISPR knockout. That would be a second thing that you would do to validate your CRISPR knockout.
Exactly.
Yeah. And even, sorry, if I can just add one more thing, even because what you say, you brought up the example of or in the CRISPR knockout on how you target one specific gene, but actually you are affecting the whole genomic locus, right? So then, even, when you say, I want to validate my knockout and use two different guides, but actually that doesn't make much difference because anyway, so two guides are not necessarily
different methodologies.
Yeah.
That's what I'm saying. And to be honest, and they're out there in the literature, you could see papers that people like targeting amplified regions and like just showing that their dependencies, like Mick and some of them are like, they're out there, and that's an important control that
So you'll be better off doing like a CRISPR knockout and then perhaps use shRNA, maybe.
Yeah, shRNA is any other methodology, but you want to,
okay, I understand.
I think my main message is always with these things that you want to multiple ways to validate your phenotype.
Okay.
Because there's no way, there's not one of these is like bulletproof.
Perfect. Yeah. Great. Thank you so much, Sefi.
Do we have more questions? Maybe I could ask one while we wait for other people's questions, but as if you saw, this is about variant classification, and if you have limited funding, how would you choose between doing base editors or scanning mutagenesis screens?
So I think that the scanning mutagenesis are more straightforward because you know exactly what the variant that you introduced in. You know for 100% that the variant was introduced, and you know the phenotype, and you got a very strong phenotype. The base editors, I think, is interesting. I think we're only starting to learn like how to interpret them. They do give some interesting information, like I showed about introns.
Yeah.
But with the base editors, we're always predicting, right? Because we're not sure that the actual variant was introduced even, right? Because we just measured a phenotype, so we didn't actually measure the variant itself. And a lot of times you are limited because you have to have the PAM sequence for the guide RNA to be able to work. And many of the times you don't have, your actual pathogenic variants are not within that region or within that, like, are not amenable for that particular, or if you're not like doing an A to a G or C to a T, if you're on a different variant, right? You can introduce it. So a lot of times you can't. So I would say if you only want to use one, I think that the scanning mutagenesis is clearer, easier, and more robust. But the base editors for sure are giving interesting things. Maybe in the future we'll learn how to use them better.
Right. So maybe genes like CH1 where there's I think an unusually large intron, or that introns in a specific genes are might be very relevant, then you would go for base editors.
Well, I think, to me, that's already personal, maybe, but to me, like, the introns are very interesting because they're very unexplored.
Yeah,
right?
yeah.
So,
yeah.
So to get phenotypes from introns are always, that's the more interesting one, I find. But so I guess it depends what you want to do with it.
Yeah,
if you want to find more novel things, I would think that the introns will give you more novel things, right? But if you want to get robust classification, you probably want to go for the scanning mutagenesis, right? That'll give you a much more robust and clear picture.
Yeah. And have you ever been able to compare outputs from base editors and mutagenesis screens on the same gene for exonic variants?
Ask me that question in about 3 weeks.
Right. I'm writing that down. Back to Sefi in 3 weeks. Right. Do we have any more questions from the audience? Maybe can I very quickly ask you, Sefi, about the MAVE? I was hoping you'd talk to us about the MAVE and how. Can you say a few words about it and how you can contribute data? Like, if we not doing these functional screens, but we want to see if anyone has done functional screens.
Well, MAVE is good for variants. So MAVE is a database that anyone who's interested in doing variant screens. So it's not for gene screens. It doesn't have in it. It's only for variant screens. So anybody interested in doing a variant screen, MAVE is a database that you could kind of input the screen that you're interested in doing, and we'll get information if anybody else is doing that screen or interested in that screen. We have actually found that way. One of our screens is that someone else is also doing them, and we're now working together with them. So, hopefully it's more productive. I think that's a good way, a better way to do science instead of just competing, to work together for a goal instead of just scooping each other, which serves nothing. So anybody who's interested, it's free to register. And if you're doing these kind of screens, I think it's very useful.
Okay. Well, thank you very much, Sefi. I don't think we have any more questions, and we are getting close to 1. So thank you to you, and thank you everyone for attending. I hope you have a very great afternoon. And for the next MPCCC events, please check out the website. We'll have a couple more tech talks before the end of the year. So thank you.
Thank you.